Now coming to the flavonoids, these are basically polyphenols because of the basic constituent phenol having a basic unit, and some of the prominent examples includes the carotenoids. Even the chlorophyll is a class of polyphenols, and they can be water soluble. They can have several colors. They can be pink, they can be red, they can be purple or blue. So majority of colors that you see in flowers and fruits is primarily because of presence of these flavonoids. And they are basically, chemically you can say, hydroxylated polyphenols with basic unit like this, and based on where hydroxylation has happened, they can be classified under these 6 classes, including flavonols, and even the chlorophyll is flavin 3 all, where it is here the position, then flavanones, then isoflavonones, and so on. Now if you try to see the examples of them, that include the berries. Berries are having anthocyanidins, and the active compound is cyanidine. Similarly, tea, what we drink, which gives us a kind of refreshment, is primarily because of presence of flavin 3 all, and the active compound is catechin. Broccoli and onion, they again have the flavanol class of protein, and the active compound that has been reported in them is quercetin. And then Sputularia, it is also an example from where this bacillin is commercially produced. It is having the active compound flavanones. This is grapefruit having asparagine, and the soybean from where this isoflavone like benzene is commercially produced. Now, third class of secondary metabolites are characterized by presence of nitrogen, and if you smell them, just from the smell you can make out that they are alkaloid class of. They have huge commercial value, and you can identify with their characteristic ending in the name with I and E. It's not uniform, it is not for all, but majority of alkaloids. Like if you take a case of opium, from which this morphine is produced, isolated and purified, that is having characteristic ending I and E. Then we have quinine, that is isolated from cinchona, again having this anti-malarial value. This tobacco that is produced from Nicotiana tabacum, what we call as, and with active compound as a nicotine, act as a stimulant. Vincristine, vinblastine, that is from the Catharanthus, have second most prominent anti-cancer compound. Then comes the camptothecin, which we will be talking in detail after this initial part of secondary metabolites and their role. So now, with brief introduction of these secondary metabolites, their commercial value, their importance in plant system, how they are important, I shall be taking you through the journey of camptothecin, why this compound is important, and why we should increase it to meet the global demand. So basically, camptothecin, as I showed in previous slide, is a pentacyclic alkaloid produced from the TIA pathway, that is terpenoid indole alkaloid pathway. And then it is a fourth prominent anti-cancer compound with global demand more than $4 billion. That shows its value, commercial value, because anything we do research or we produce a technology or we start a startup, unless it translates into commercial viability, that product will only remain limited to the what we do in lab. The value is primarily because it's anti-cancer compound, and the derivative of this compound, which are used in medical field, is used for the colon, ovarian, and the small lung cancer treatment. And if you look at the natural source, till now our dependency for this compound is only on natural source. You may ask that why can't we synthesize it chemically? We can discuss this later also, but just to brief it, the biosynthesis of this compound is so complex. Even if we are able to demonstrate their biosynthesis chemically, that means pure biosynthetic in lab, the two aspects that comes into picture, one thing is the bioefficacy. That means the end product that is formed after doing several purifications of cycle is having that much bioactivity as the natural ones, because let us accept the fact that we cannot match the natural engineering capability. Second thing comes the cost. As long as natural sources are available, whether from the directly from the natural presence or from the tissue culture means, if we are getting the source of this material cheaper than what is the value of commercially synthesized one, we have to depend on the plant source only. And here comes the three plants, Camptotheca acuminata, that is primarily grown in China. Metabolites, Cordida, and Ophiorrhiza are from the Indian origin plants. And they have the CPT concentration in a range of 1 to 3 grams per kg. So that is the limitation. That is the limitation with most of the secondary metabolites. They are synthesized in very low amount. To elucidate how important this compound is, I will give the cost, that how expensive, that's why chemotherapy is so expensive. These values I am giving in terms of some values in terms of US and some value even in context of India. Like the commercial one, a cost of 10 tablets of 1 milligram each sold by GlaxoSmithKline, that is sold as a Hicamtin, is around $4,200 US. So you may calculate almost $420 US per milligram of commercial topotecan that is formulated using this camptothecin purified from the compound. In India, the generic one, the topotecan, which is given during the chemotherapy, costs something around $25 US. That means around 21. This previously I have calculated. You may calculate with today's US dollars rate. In injection, irinotecan, that is another generic version, another derivative that is produced from the camptothecin. The cost of injection is in US is $1 US per mg, sold by Pfizer, and the commercial name as a Compostar. So this shows how much the cost of these, or the expense of these drugs comes when we go for the treatment of various cancers. Now, why we need to go for the research and why we need to go for the engineering, what we call as a metabolic engineering. Metabolic engineering is basically, if a pathway is happening, here I don't have slide, but we will discuss in next slide what is basically metabolic engineering. But in this slide, what I want to convey is that the natural resources, these compounds which are having so much of commercial value, their label in natural source is very low, as low as 0.1%. That means if you take out the plant, dry it, make a powder 1000 kg, from the 1000 kg you may end up with 1 kg. And that's why the cost of these compounds goes so high. And also cost comes the purification and formulation. And many of these plants grow very slow. You take a case of Mapia, the 0.1 to 0.3, what I told, that means 1 to 3 milligram per tissue. That value reaches when a plant attains a maturity of almost 13 to 14 years. So if a farmer grows or any progressive person grows those plants, they have to wait for almost 13 to 14 years, and then only that much level of that compound we will get. And here the Camptotheca is having little advantage because the Camptotheca grows just like tea. So upper part of that leaf we can keep on plucking and we can keep on using for our extraction purpose. Many of these plants are endangered list, including the Mapia in India. Legally or without declaring, we cannot cut even a single plant of Mapia, which are primarily present in Nilgiri region, right from the Maharashtra up to the Tamil Nadu and even a part of Kerala. Why they are again expensive? Their total chemical synthesis is complicated, costly, and finally not economical. So as long as the process is not economical, most of the pharmaceutical companies, what they do is they either cultivate them or they try to get it illegally. Like in, I don't want to put into the record, but in India also what the people do when they want to sell it illegally, they will cut it, they will label it with some other name, and then they will sell it to some of the European market or even including China and Japan. So there they make powder and they sell it at very high premium price. Now global demand for these compounds is increasing day by day, and with limited supply, the cost is going on higher side. To show you the example why these are having so high commercial value, you take a case of Taxol that is produced from Taxus peccata. The market price is close to 150 to 160 US dollars per kg. And what is the ratio of this compound? If you have 30,000 kg of this powder purified from this plant, you will end up with 1 kg of drug. So ratio is quite low. And global demand is more than 13 billion US dollars. And the drug that is formulated from this compound is paclitaxel, the number one drug in terms of commercial value across the anti-cancer compound. Second is Vincristine vinblasting. It is one of the most common plant grown even in India, what we call as a Catharanthus rhodius or Sadabahar that grows in most of our gardens. But there are certain varieties which are having high level of this compound, and also because of availability and faster growth being a herb, the overall market price is relatively low. And the compounds, the commercial products from this compound includes Uncovin and Vincasser. Now, third most prominent compound in terms of global value is camptothecin, that is purified from these three sources that we just explained, and the market price is close to $2,500 per kg, and ratio is relatively higher. You can say almost 1 to 3. I gave examples. So 1000 kg, if you have dried powder, you will end up with 1 kg of this drug, and market value is close to 4 billion. Here I have not updated. And the commercial product from this compound includes Topotecon and Onotecon. So at our lab, we are working on two plants, Metabolites foetida, which is a tree species, as I indicated, grows to a maturity height in year of 13 to 14. And second one is your Ophiorhiza. Several species of Ophiorhiza are present, and what we work on, one species is Rugosa and another is one Mungos. So these two species we are working in our lab. So what we do is, along with the tissue culture approach, because I indicated we want to decrease the dependency of our these sources on the natural one, so that we can provide an alternate. So that's where the tissue culture can come, which produces the clonal progeny of similar type of plants in a very limited time. Then metabolic engineering. We have just started almost 3-4 years back, that apart from using our conventional approach of tissue culture and allied things like hormone combinations, we can try to increase the yield. We can try the different elicitation method to increase the yield. And then comes the metabolic engineering. So we try to explore whether this metabolic engineering can be used in an additional way to improve the CPT content in host plant. So why we call it as a metabolic engineering is, in this secondary pathway through which these secondary metabolites are synthesized, many of these secondary metabolite pathway are having single precursors. So you may take an example that if a product form B, so B can go to almost 10 or 20 directions. It can go to C, it can go to D, it can go to E, like several fates of product B can be there, here can be there. But out of those 20 or 30 different pathways where a substrate can go, only one pathway gives our product, which is having high commercial value. So metabolic engineering is nothing but to divert the substrate into the direction where our product of interest, which is having high commercial value or high economical value, should be diverted. So different ways to do that is, you may block the other pathways where substrate may go, or you can increase the rate of reaction which take the substrate to the pathway of your interest. Like if you take a case of this camptothecin biosynthesis, it is a merging of two pathways that includes MVA pathway and shikimate pathway. These both are primary pathways. So the products of these primary pathway, when combined, the product of this is streptamine, and whereas this is having shikologenin. So when they merge, they form the streptozidine. So this streptozidine is a kind of precursor for an array of secondary metabolites, not just camptothecin. In Catharanthus, it will form the Vincristine, vinblastin, ajmalicin. In Metabolites foetida or in the your Ophiorhiza, it will form the camptothecin. And there are several other pathways. It can be also used for the several other secondary metabolites biosynthesis. But what we want is that the streptozidine that is synthesized at this step should go only to this destination. And that's what is metabolic engineering is. And some of the putative enzymes that has been identified and their corresponding gene later working are listed here, like granule synthase, granule 8-hydroxylase, 8-hydroxyl granule oxidoreductase, and STR. That is story of this STR I shall be sharing just in a while. So overall approach is something like that we shall identify the candidate gene involved in the CPT biosynthesis using comparative de novo transcriptomic analysis. Why de novo? Because there is no reference genome available for the comparison. Characterization of candidate gene, that is the overall aim. Then we try to engineer the cell culture, hairy root cultures of target plant. Here I mentioned Ophiorhiza rugosa to finally increase the CPT yield, and then take up that to a optimized or the standardized bioreactor thing. That is how our approach is, or even for that matter, even if your professor gives a problem that you have to solve this, first we have to see for the approach, and then we have to solve one by one. So this slide is very important. When we want to identify a gene, especially if nothing is known about the system. See, with advent of many genomics technologies, the cost of sequencing of genome has drastically come down. But again, it depends upon the need, and it depends upon the how many research groups are working across the globe. If you take a case of Ophiorhiza, if you take a case of Metabolites fordida, though they have a high commercial value, the number of labs across the globe working on that plant are bare, I can let's say less than a dozen. Like into this aspect, not only purification I'm telling. So when number of labs working on that, the number of resources available for us to explore the papers, the references are also less. So what we started doing is what we call, or what you are taught also, is called as a differential gene expression analysis, DERs. In older times, there were other classical way what we used to say as a colony hybridization. You may try to revise in your basics that what the colony hybridization used to be, or differential expression analysis. So purpose of differential gene expression analysis is to compare the gene expression analysis in two distinct tissue samples. Now here, our objective is to understand how camptothecin, an important secondary metabolite having high commercial value, is synthesized in Ophiorhiza plant. Like if you take a parent plant that is having 0.1 dry weight that is reported. So through tissue culture, we have developed the different developmental stages of this plant, and we also analyze the CPT present in them using the HPLC. Like if you take the fibril callus that is produced from that is having traces or non-detectable level of camptothecin. When this start little differentiation, where is the root primordia have started coming, it is having 0.009% dry weight. And then more prominent root comes, it increases to 0.03% dry weight. So from here to here, journey is almost 30 times. So this shows that this has something to do with the differentiation. And just to put things in perspective, if the same hairy root that is developed from same plant is having almost 0.3% dry weight of camptothecin. So any work that we have to start with the differential gene expression analysis, we should have the tissues which are having the character totally different for our objective. Like here traces, that means camptothecin is almost nil, and this is where the camptothecin is 30 times more. So what we did is we have selected these three tissues and went for the differential expression analysis. So how it is done is, what you do, you take the samples, purify the RNA, and then convert it to cDNA, and then go for the sequencing. Now, after all the quality parameters, we check how good the RNA is, how good our cDNA library is, then start the process of sequencing, what we call as a next generation sequencing. So before coming to the next generation sequencing, I will illustrate what our earlier approach, then you will appreciate. You must have read about the Sanger sequencing, the classical way of sequencing our DNA fragment. So that is primarily based on your dideoxynucleotide. So because your deoxynucleotides are having your hydroxyl group at 3' end, if you remove that, the chain extension won't happen. So where the dideoxy is added, and that base used to be labeled. So based on where this reaction has terminated during the PCR, your product formation also used to terminate, and thus these products which are formed after the PCR were loaded onto your gel that we call as a classical Sanger method, and based on that you used to get the sequencing data, like the products of different size, like here dideoxy T is added, here dideoxy G is added, and each base is having different colored fluorophore molecule. That means that will give different fluorescence molecule when that product passes through the detector. So based on the fluorescence emitted by the product form, the system analyzes and then give a final data, something like that. The biggest product is having C in the end, then the one base shorter fragment is having T at the end, the even shorter has G, and so on. So that's this you translate into the complete sequence what you want. So here if you observe this analysis or the data you start getting only after the entire reaction in the first step happened and this total product was loaded onto a gel or into the capillary. Now what has happened into the NGS is different platforms are there for the NGS. You try to only limit to the Illumina, which is one of the most prominent. Illumina and Torrents are the quite the common ones, but Illumina HiSeq I shall be discussing because that is used by most of the companies which are based in India, including one in Cochin, one at Hyderabad, one and majority in Bangalore, where they do the sequencing of these samples. So what we go back, what we did is we have isolated the total RNA from this sample. We have isolated the total RNA from this sample.
Excuse me, your slide is not visible.
Now?
No, your slide is not seen. Your slides.
Yes, sir, it is visible.
Is it?
I think there is a technical issue.
No, I cannot see
with
the
your
slides.
slide.
So is it visible now?
No, not yet. You please check it. No, in my system, I cannot see the. Okay, you please continue. Okay.
Yeah, okay. So what has happened is in this platform, the term used is sequencing by synthesis. NGS is also called as NBS. That means the sequencing will keep on happening and you will keep on observing or capturing the data. It will take a lot of time to explain this technology, but just you in brief, I can tell you what happens exactly. Like here you have got, I explained you, we have started the RNA from the 3 different samples which are having differential level of camptothecin. That RNA was converted to cDNA, what we also called as the DNA by the reverse transcriptase. And this sample is then fragmented into small spore because these reactions cannot go beyond their capability. Some are 50 bases, some are 100 bases. That depends upon the platform use. So if it is 150 bases, what you try to do, you try to fragment your cDNA into as small as 300 base pair that need to be optimized. Once that is done, you have a kind of flow cell. Flow cell is, you can try to see miniaturize your ELISA plate. It is basically glass plates where so many spots having the adapters are spotted. In earlier times, we used to see the microarrays, something of similar sort, but very miniaturized. If you try to understand, you try to recollect the ELISA plate where you have 96 well ELISA plate or you have 16 well ELISA plate. So you try to exponentially increase that millions of wells are there onto this flow cell. And each million of cells will have single reaction in them. So now you try to visualize in Illumina based sequencing, almost 1 million sequencing reaction happens parallelly and millions of bases of that data is generated after every round of synthesis. So what happens is these are your spots where the adapters are present and this fragment is your cDNA having the adapter at both the ends for the binding of these sequence to these matrix, and that's where your sequencing like this happens. If your adapter, this cDNA binds here, the sequencing will happen like this, and if it is opposite one like the blue one bind complementary to this, the direction will be like this. So synthesis happens if you take a here, this is adapter, this is your cDNA sequence. So what will happen if complementary base here is A, then it will synthesize T, then T will give particular. So after addition of T in this particular cell, it will give green color, and this particular, assume this is reaction number 1. So this particular flow cell will emit E, green color, and accordingly it will be coded that this well is having T base. Similarly, all these wells will be keep on counting by a very high resolution camera for the image acquisition and for the analysis. So after that G will come, like G has come here. Same positions, second round of synthesis. Same position, third round of sequencing. So what happens is, here first synthesis happens, you capture the data of the fluorescence, give the treatment, this fluorescence is lost, then again add the second round of, do the second round of reaction, second base is added, again you capture the data, again you give the treatment, fluorescence is lost, again third round, so keep on going, almost 150 rounds of reactions, or based on the platform, it can be 50 by 2 or 100 by 2 or 150 by 2. So those many basis sequences comes, and that is first step of sequencing is completed. But given the technological advancement, this is one of the easiest thing I can say. As a researcher, the most important trouble starts when we get so much of data. I just showed you the millions of reactions are happening, and you can multiply with how big your sample is. So here comes the analysis. So analysis is in terms of the sequencing results that you have got. You have to make meaningful information from that. Like you have got a huge stack of sand, and from that you have to find out your information of interest. And here my interest was to find out the genes that may be involved in the biosynthesis of camptothecin ophioryza. So because here reference genome was not available, if you try to recollect, when most of you must have done the BLAST. So in BLAST, what we do, we have something to compare with. We give the reference thing, and against which we BLAST. Like if you want to give the specialized BLAST, like you try to search my sequence of interest only into the human genome. Now human genome is completely sequenced. So what the BLAST will try to do, it will search for the similarity of your sequence or the query what you have given only against the human genome. But in case of ours, neither MAPIA genome is available, neither ophioryza genome is available. So what it tries to do, what the pipeline, what we use is called a de novo. We may try to, if anyone is interested, they may read this very beautiful paper, a Nature Protocol. It is an amalgam of biology as well as bioinformatics, biostatics, and so many things using Trinity platform. So what it does is, this platform is having 3 steps where we get the raw reads after the NGS. We have got the raw reads. So what the system inchworm, that is step number 1, will try Trinity, because this involves the 3 steps. So first step, what it does is, it removes the adapter sequence, the non-redundant or non-relevant RA sequence, which doesn't qualify a certain value, and then gives the pure RNA reads. And then the similar kind of RNA reads. See here, you try to understand that if you take a case of human, we are having only 30,000 RNA, functional, that means functional RNA. So you should get only reads 30,000, but that is not the case. There will be many pools, so same cDNA or the same RNA must have got sequenced several times, but all of those sequencing reads will be similar or exactly same. So based on the similarity index, those sequencing read will be pooled and what we call as a linear contigs. They will be pulled together and they will be clubbed as a single. Because these many sequencing results are similar, they are pulled together and they form the one. And after that, these contigs, you try to fill in the gaps and form the what we call as the de Bruijn graphs. That involves a lot of biostatistics, many cutoffs. And then you try to compare the samples, like the one which has got very low CPT and the one which has got high CPT. Those transcript you compare that what is the fold change, what is the isoforms, and so on. And it involves a lot of your hardware requirement. To just elucidate, if your plant sample, if you are asking for almost 35 million reads, like in our case, I can give you example. If I want to get 35 million reads from my samples, the system where I need to process my sample should have at least 1 GB RAM per billion reads. That means if my sample is having 35 million reads, the PC must have a RAM of close to 35 GB. Most of our laptops, most of our PC are having RAM as high as 16 GB or more. So if you put our data into normal PC, our PC cannot run simply. Many more other software required like Course, Putty, Meg, Amber, and other like sudo apt-get. So we can skip it. It is a very bioinformatics people, they can end. Just to appreciate how we get the results. So at the end of the day, what we'll get, we will be given, the sequencing person will give us a file of as high as 12 GB or so. So this 12 GB data we have to process and make out our meaningful information. And these are some of the features. So just I want to show that how big files and how much data is generated at the end of NGS. Now to cut the story short, what we got at the end of analysis of two tissues of MAPIA, or here Ophioryza I can say, one or the total were around 178,963. Upregulated transcripts in both samples were 46. We have to get the meaningful transcripts which may be involved in the biosynthesis of camptothecins. So by narrowing down this entire story, and this is how those transcripts are again classified based on their similarity with the available database. What happens after we get the finally processed data, we try to convert that to amino acid, and then we do the what we call as a bulk blast against specialized databases, and then on entire pathway. Like this one is ethanoate synthase. Here comes your tryptophan synthase, streptozidine synthase, then geranyl 10-hydroxylase. These are some of the enzymes which we could narrow down from this pool of information what we got after the struggle of close to 6 months to 1 year or so. Now, once we have got hold of these putative sequences, which may be these genes, the story begins that we must confirm whether these are same gene, because these are only in silico data, only the data that is given by NGS, whether that is happening in real samples, we must prove that to complete our story. So I will just show you the story of STR, that is streptozidine synthase, because that was one of the crucial enzyme combining tryptamine to shikologanine to form a penultimate precursor for the biosynthesis of camptothecin. We have also worked on this, but here I shall be sharing the story of only STR. So what STR does is, it does the condensation of tryptamine with shikologanine to form the streptozidine, and our sequence identification cDNA showed a sequence of 1059 base pair. So what we did is we cloned this overexpressed in bacteria and we could see overexpression. Then we purified this protein. Once you have got the hold of protein, we can only confirm the functionality part of this by artificially supplying these two substrates and adding other cofactors, and then putting our purified protein and see whether this compound can be synthesized or not. So basically, in any enzymatic reaction, I have given A, B, and see whether product C is synthesized or not by adding this my protein of interest, and that was doing the job. That means that the sequence that I have tagged as a STR was indeed a streptozidine synthase. It was able to combine these two products to form my product of interest. This we again proved in the tissue whether it has something to do with the camptothecin levels. And here, as you can see, your wild type leaf, this data I am showing with respect to MAPIA or another plant. So in MAPIA leaf, the level of camptothecin is among these three tissues less, followed by roots, and maximum in immature seeds. But we could not find much difference in respect of our STR, that streptozidine synthase gene expression. But so that means here the expression was not directly proportional to the CPT level, because these are kind of high level of CPT is present. But same thing when we try to do in the callus, the regenerated shoot and the regenerated small plantlet. So I have shown also that in callus the biosynthesis of camptothecin was minimal. What we observe similar things even in the case of camptothecin. Here you can see camptothecin is not callus doesn't have camptothecin. In regenerated shoot, only traces of camptothecin, whereas the regenerated shoot along with the root of MAPIA has shown an amount of camptothecin. And here the relative NFSTR expression was quite correlated with the level of your camptothecin. That indicates that level of NFSTR during the developmental phases of process is directly proportional to the CPT. So with this hypothesis, we try to overexpress that in our Ophiorhiza, and that's how it's generally any transformation is done. You select the explant, do the transformation, develop the transgenic plant, and look for the camptothecin level in the final product. And after PCR confirmation and southern hybridization, we have analyzed the camptothecin in these plants, and the best 5 lines we have analyzed, and these 5 best lines have shown significantly improved level of camptothecin compared to the control non-transgenic plants. So these all plants are plants that are overexpressing the streptozidine synthase gene. The best line has shown close to 1.9-fold higher CPT compared to the control non-transgenic plant. So this is how the story goes, where you start with a scratch, like you have identified the gene, you have characterized the gene, you have assigned the function, then you try to prove the role by overexpression. Now, here in this case, this may result in the increase in camptothecin, in other it may not, because majority of these secondary metabolites biosynthetic pathway is multi-step process. Like if 10 steps are involved, just by altering one enzyme, we may not get the product of interest at higher concentration. That we need to check. If by increasing the flux of 1 or 2, that's why what we are also doing is we are trying. So overall story of this previous slides is that what we did is we are increasing the expression of STR. So more and more STR enzyme is formed. So more and more tryptamine and chicologenin can be converted to streptozidine. More and more streptozidine lead to the more and more biosynthesis of camptothecin. Now what we are trying to do is further, apart from characterizing the genes that are involved here, like I have mentioned geranyl-10 hydroxylase here, tryptamine synthase here, we are trying to see the various transcription factor. If you try to recollect, in majority of cases, transcription factor are a kind of master regulatory things. They control many steps. So single transcription factor, like you all know ORCA3 class of class of proteins that controls almost 5-6 genes in one shot. So if you overexpress ORC or similar kind of transcription factor in our target plant, you may end up in the overexpression of many genes, and in turn, more and more biosynthesis of CPT. So these are the different steps of how we produce the hairy roots. We infect the hairy roots, and that's how we get the hairy roots. And hairy roots are beautiful systems because here pigments are less without compromising the amount of camptothecin synthesized. And in fact, the transgenic hairy roots have shown 3-fold higher camptothecin compared to the parent plant. Now these are other things. We have taken this further to the flask and finally the bioreactor, wherein we try to grow the hairy roots at a bulk level and see how much camptothecin they are producing. And in fact, we have realized apart from producing, they are also leaching into the medium. So medium as well as these plants both will act as a source of our camptothecin. So this entire story I just summarized in single slide. Like what we did is we have overexpressed the STR, and this was our control plant that is having 0.1 percent dry weight camptothecin, and by transgenic approach that is overexpressing STR, we could achieve 2-fold higher CPT in transgenic plant. And when we overexpress this STR in transgenic hairy roots, we could get 3-fold higher CPT in transgenic hairy roots overexpressing the same STR gene. That is our transgenic hairy lines. Our protocol with respect to another plant that is Mapia is also ready, and we are also doing transformation, but you must remember that Mapia is a tree species. So if you are given a choice to work on a herb species like Ophiorhiza, and you are given a choice to work on tree species, it is always advantageous to work on herb, because tree species, though they will be having final level of camptothecin higher, but they are difficult to regenerate, they are difficult to transform, and they take longer time. Like from here to here, the entire process take as high as almost 1.5 years. Where in the case of Ophiorhiza, right from here to here, it takes just 12 or less than 2.5 months to 3 months time. So that also makes sense that which system to select. In the end, I shall be just sharing the couple of technologies available for the startups or the entrepreneurs who want to take. One is the mass propagation protocol of Ophioryza rugosa. That is the plant that we are working as a source of anti-cancer drug for the camptothecin. And this has been transferred to 4 companies, including Patanjali. Recently, we have also developed the micropropagation protocol for turmeric as well as ginger, based on the input from the farmer, where they want to use this as a substitute for the rhizome for the cultivation to get the disease-free plant and save the time of cultivation. This is also available for transfer. In end, once again, I thank the UC College. They were kind enough to invite me and share my research with you all. It is very important to give a feeling of research and how we can motivate the youngsters. And I personally would like to congratulate your department to take such kind of initiative to the larger part of students, which will encourage them to take research as a career. I am equally thankful to Professor Shireen, who was coordinating everything and inviting me for this talk. And a special thanks to my mentor, and still I consider as my guide, Professor Epan, my senior colleague, who is, whenever I have any issues, I always take her suggestions. So thank you very much. Now I am open for the questions, and whatever questions are there, I will be more than happy to answer them. Thank you.
Yes, sir. That was an amazing virtual treat for all of us. Thank you, sir, for such an informative presentation. We are grateful to you for the time and effort you took to share your knowledge and lecture with us. I hope you all enjoyed this session. So now, Dr. Sudhir, sir, will answer some of the questions that we received during the presentation. So, sir, here is the first question. In India, we have various alternative medicine systems like Ayurveda and homeopathy. So do you think secondary metabolites are responsible for their efficacy in treatment?
They are definitely. I must appreciate that India is fortunate enough, and equally is China, to have such a large, vast information about these plants and the various remedies based on these plants. The problem with us is why it has not gained so much of acceptance, especially in the Western countries, is that we don't know the bioactive compound present in these products. Like we may be using any herb powder, we may be using a formulation based on a combination of plants, and that may be working for us. But if we are asked to prove that which compound is basically giving this function, whether if person has got a fever, we give some kind of herb to them. If a person has got malaria, we'll give a kind of powder. But if a certain scientist or some person asks what is the active principle of that herb or powder which is doing this job, unless that is solved, the acceptance in Western country or the overall global acceptance of these remedies will be limited. Second thing, what comes with these products is quality. A same plant that is grown in Kerala and another same plant is grown in Himachal, the content of, apart from our product of interest, other content, be it heavy metals, be it other metabolites, that may be having some deleterious effects also, is another question mark. So quality control is also an important factor that we need to work on. If we can answer these two things, that is bioactive compound and quality things, I am sure these products and the plant-based products will have much, much wider reach across the globe and not just in our India and these countries.
Dr. Sudhir.
So here's the...
Yeah, I am Dr. Satish Kumar. I am a retired scientist from Botanical Garden. I worked on this camptothecin and clonbagin nearly about 30-35 years.
Nice to know, sir.
Yeah, in fact, I had a collaborative program with your senior colleague, Dr. Raja Gopalakrishnan.
Yeah.
Beginning, Dr. Susan mentioned about that.
Yeah.
So I would suggest you that instead of this Ophioryza rugosa, I think Ophioryza mangos, because the project was on selection of desirable different species of Ophiorrhiza with maximum camptothecin content. Okay. So we found that this Ophiorrhiza, and we have collected nearly 19 species of Ophiorrhiza through Kerala. Okay. And we have come to know that there are 4 species which are very potential with respect to camptothecin content. And among these 4, Ophiorrhiza mangos contains the maximum amount of camptothecin. That is nearly, I remember, if that is 0.6%, 0.4 to 0.6%.
Okay. Excellent.
And maximum amount is in the shoot system, that is during flowering.
Okay.
Okay, and that also we have studied at different stages of this plant growth. And we have published a very good number of publications in good journals. And so it is better you take this Ophiorrhiza mangos, that is a very potential species. And what is the growth rate of this hairy roots in your shade plant cultures or in the reactor level? What was the growth rate?
Yeah, it is having doubling time close to 6 weeks.
Close to 6 weeks.
Yeah, but one thing I can share on this platform is the hairy roots that grow in liquid, the growth rate is not that fast compared to when a solid substrate is poured.
Okay.
That is a general observation.
Yeah. But it is very good if you could take this cell suspension culture. They grow very well. And we have published one paper in Biotechnology and Bioengineering about this cell line selection and also using some, elicitors. So that would be a wonderful choice. Anyway, it was a wonderful presentation. So I congratulate you. So I hope we can meet.
Thank you, Dr. Krishnan, for your very nice suggestions. In fact, our transgenics works, we will definitely take your inputs in terms of plant selection, in terms of systems. Anything in case of self-suspension, what we observe is when the system is kind of non-differentiated, the level of the camptothecin produced is quite less compared to what we get in differentiated. But
yes, that is true. And one more information I would like to share with you is that this multiple shoot cultures, that is also a very, very prospective system.
Yeah, that's true.
Yeah, cell suspension cultures, they grow very well, but camptothecin content is very, very poor, very less.
Yeah.
And coming to this Mapia, this Metabolites pneumoniana, that's a very difficult species. You must be very careful when you take that material. Earlier Dr. Roja was also working on the species.
Okay.
It's a very tough material.
I agree.
Yeah, all these Icacinaceae species, they are totally recalcitrant to a certain extent.
Yes.
And one of my students, she has submitted her thesis on Pyrnacantha volubilis. That is another wonderful species which contains camptothecin, which is much more than even Metabolites and Ophiorrhiza. That's a lioness species. And it's also very difficult to propagate plants under in vitro conditions. So you must be very careful when you take this plants belong to this Icacinaceae family. Ophiorrhiza is a wonderful choice. You can work on that. Anyway, I congratulate you. It's a wonderful presentation.
Thank you, Dr. Krishna.
Satish, can you give some Ophiorrhiza mungos plants to Sudhir?
No problem, madam. No problem. TBJR now at present I don't have any connection, but if he comes, I can share some of these materials and we can collect from the wayside to Ponmudi.
Okay.
No problem.
Thank you.
Thank you, Dr. Krishna.
Okay. Thank you. Good.
Thank you, Krishnan sir, for your contribution. So here comes the second question. Are there specific exclusive databases which give structural and functional information on secondary metabolites?
Yes, there are specialized databases and I can share the website also. There are numerous websites. One website may not be having the list of entire secondary metabolites. Like some may be classified as a flavonoids, some may be classified as a terpenoids. But yes, there are websites wherein the list of secondary metabolites, especially the ones which are having high commercial value, is very much available. And I can share the same on the whoever want to request.
The next question is, can microarray technology used for differential gene expression analysis?
Yes, definitely. The overall purpose of microarray was same, but what has happened with the advent of this genomics technology as NGS platform, this microarray has become obsolete. I won't say obsolete, but given a preference to a researcher, the people will prefer to go for the NGS than going for the microarray. Because the limitation with the microarray, you have to prepare the microarray profile for the plant of your interest first. You cannot directly go without the availability of microarray, because there should be some standard against which you will do the comparison. That is the only thing. But yes, if you compare in today's time, if 1,000 researchers are given the similar objective of differential gene expression analysis, I can say more than 75% will prefer to work with the NGS platform than the microarray.
With reference to the slide with metabolic engineering, how do you trace all the pathways leading to formation of array of secondary metabolites?
As I indicated, we were primarily interested in the biosynthesis of camptothecin. And this camptothecin biosynthesis is not at all still clear with 3 plant systems which are producing this camptothecin, be it Camptothecin acuminata or Uchorhiza, or even for that matter Mapia. Only whatever is given is putative. That based on the number of so here if I try to explain you how these pathways are elucidated, what you do is you purify the products from this from the tissue, or you do the precursor feeding with labeled C14 thing. So if you are giving the carbon source as a C14, what you will do is at different time interval you will isolate the products and you will try to do the MS analysis of your products. So based on the product profile which are having similarity with the camptothecin biosynthesis, it has been proposed that the pathway will be something like that. But there can be a condition where some of the intermediates are so short-lived, they synthesized only for a few seconds and then get converted to their next product, that during the process of isolation and then followed by MS analysis, they are lost. So I have whatever the pathway I showed is only putative. It is not exactly that camptothecin biosynthesis in Ophiorhiza or Mapia or Camptothecin acuminata is working exactly like this. But based on the research inputs across the globe, the people have postulated or people have proposed that yes, camptothecin in this plant may be happening like this. But there are again many fill in the blanks. They know from the A to C synthesis has happened, but what was B they don't know. But so what they will do, they will in the pathway they will only show A to C conversion. In between B, B1, B2, B3, or many steps were there that they are not aware. So this pathway is only based on those inputs, and it is not exactly the way this plant is happening. Now coming to the selection. In what happens is other pathway, if we don't know, obviously we cannot block. But yes, if you take a case of strictosudine, that may go to 3 pathways, say for example, and we know which are those 3 pathways where this strictosudine can go. So what we can do is the 2 genes that do the conversion of strictosudine to the other pathway, we will block using knockout strategy or by RNA interference strategy or microRNA based strategy. So if those 2 pathways are blocked, our strictosudine conversion will be directed to only this camptothecin biosynthesis. But this is again hit and trial. In plant, things are not that easy as they are for the mammalian system or even for that matter bacterial system.
How to do identification of same RNA?
Same?
RNA.
Same means which like RNA?
Yes, sir. That was the question being asked.
So same RNA, I assume that the person want to ask the characterization of any mRNA. So if RNA you have in hand, the simple step you have to do is you have to convert it to DNA, because the stability of RNA is a big issue. So in any characterization of mRNA, what you need to do is first purify your mRNA of interest. That is most difficult job. Once you have got hold of that, at that table basically it is not purified. What you do is you purify the total RNA, convert that to cDNA first, and then you start doing the identification part. So here, like in our case, we were not aware which RNA is responsible for STR. So we have done the sequencing of entire RNA that got converted to cDNA. And from that, when we did the blast of this sequence that we have got after editing and all the processes, the sequence that was showing the match with already strictocidin synthase kind of genes, like STR sequence before we reported, there are reports in other plant system that this is the sequence in Catharanthus, this is the sequence information in Camptothecus acuminata, this is the sequence report in another Ophioryza. So our unknown RNA sequence, what we did is when we did the blast, it showed the similarity with some of these reported streptococcus synthase genes. So based on that, we have postulated that yes, because this sequence X is showing similarity with the reported STR gene, probably this will be the STR gene. So based on that, we go ahead with the overexpression and characterization. So one has to go step by step. That's why the bioinformatics and in silico work helps to narrow down your overall wet lab experiments.
Here's our last question, sir. Is there any scope in researching the field of marine and aquatic plants? Do marine plants have vastly different metabolites than their land counterparts?
Definitely. I must say marine are a good source of many formulation, especially in field of plants, are coming using these marine products. Now the problem with marine, what happens, the simulation of the condition in which they grow in their natural habitat. Many of these marines grow at very high pressure conditions, very diffused light conditions, very high salinity conditions. So salinity conditions you can definitely simulate in lab, but when it comes to high temperature, high pressure and diffused light conditions, sometimes the product marine algae is synthesizing its natural habitats may not be synthesized in lab condition. But yes, if we succeed in that, they are very good source of many of the secondary metabolites. In fact, that is one of the hot field coming up in many formulations, especially for the farming field, because they synthesize a lot of plant growth regulators, combination of plant hormones, and many nutraceuticals. Many algae are eaten directly for the health benefits. So that is a big field. In fact, a lot of research is going into that direction also. This biodiesel, many are working on those blue, green alga as well as alga that can be a source for the biodiesel productions. So yes, it's a big thing. Only thing is, big things doesn't come just like that. We have to put a sincere efforts and funding in that directions, and then only it will translate into something practical and feasible.
Fatima, just a second. Dr. Sudhir sir, I think you have given a comprehensive account, and I would like to congratulate you for delivering such a good lecture, and it would be very helpful for our new students. I'm planning to have a discussion on your talk in the next class that I'll attend for the biotechnology students, because I believe that you started from the very basic, and you have come up to how the analysis, the NGS and the Illumina platforms are going to help in research. So I would like to thank you for being here, and we'll have an official vote of thanks. But before that, I'm so excited to be a part of your talk, and I think even starting from conventional biochemistry, metabolism concepts, basic botany, you have delivered, and then going through the plant biotechnology part, the journey was very exciting, and a final culmination in the bioinformatic analysis part was also crucial. So it was very nice to hear talk. Thank you, sir. Thank you for being here.
Thank you, Shyam sir, for your valuable contribution. Sudhir sir, it was a pleasure to have you with us. Thank you so much for your contribution.
Thank you.
This concludes the webinar. Heartfelt thanks to all for attending today's session. I hope it was an informative journey throughout. Now, I invite Dr. Sareen Sarah Ma'am, Faculty Coordinator, UC College, for the vote of thanks.
Yes. Good afternoon, all participants and faculty members. So, we have come to an end of this session. So, the Department of Biosciences, Union Christian College, As you know, we are always being in the forefront, conducting research activities, webinars, etc. Since September onwards, we were organizing national and international webinars, and we are very glad that we could present before you most eminent speakers who are well versed in their subjects. So today also we are very fortunate to have Dr. Sudhir Singh, senior scientist from the prestigious Bhabha Atomic Research Institute, Mumbai. So it was indeed an informative, very well packed, fruitful session. So the Sudhir sir, you presented the complex topic, secondary metabolites, metabolic engineering, also the various platforms, and all the aspects in very short time and in a very comprehensive and cognizant manner. You presented the topic, and I think all have benefited from this session, and hope to have more collaborations with you soon. So thank you so much, Sudhir sir, for accepting our invitation and joining with us today for this webinar with your valuable insights. It was definitely a fruitful session. Thank you once again. And I would like to extend my gratitude to Dr. Susan Epen. I think, as you know, Sudhir sir is Susan ma'am is the mentor of Dr. Sudhir Singh. So she has been very instrumental to bring all the resource persons. She contrives the best resource persons to us, and she provide tremendous I mean, for providing tremendous amount of knowledge to the student as well as the teaching community. Susan ma'am was always in the forefront, and thank you ma'am for bringing us most eminent persons throughout during this webinars. And thank you once again, and I extend a warm gratitude to you ma'am. And in the absence of a principal and manager, I propose my sincere thanks to them, because they are always being the strong pillar for all our endeavors. And I extend my heartfelt thanks to Shyam Mohan sir for being the perfect support, guidance, and all. And he is being the catalyst that stimulated us to do the best. Thank you, sir, also for giving us the opportunity to provide the best. And also I'm thankful to my colleagues and students who took pains to throw such a wonderful event. And my special thanks goes to Fatima, and also Fatima, who compared this session, and also to my student Mr. Ashwin Krishna. He gave me all the support to do the registration and as related activities. And all others who have contributed to this session, I thank all them. And without the active participation of the student as well as the faculty and the teaching professionals, this webinar would not have been a great success. So I place my sincere gratitude to all the participants who have attended the webinar, and thank you for being with us always. And see you soon, see you next week with one more fruitful webinar, that is from by BC Velayudan from James Madison University, USA. So she'll be delivering a lecture on gut microbe and all. So that is next week, 16th. It is scheduled on 16th of evening, 6:30 p.m. So hope to see you for the next webinar. Thank you all for your valuable suggestions and all. Meet you soon. Thank you. Thank you, Sudhir sir, once again for giving us a best. Thank you, sir.
Thank you, ma'am. Here's a gentle reminder to all the participants out there. The feedback form posted in the chat box will be accepting responses. E-certificates will be provided only to those who have registered and actively joined the session. Thank you.
Thank you, Sudhir. Thank you. Azareen, thank you, Fatima, thank you, Shyamohan, thank you all the others who participated.
Thank you everyone for
There was
nice
some problem
interaction.
in between. I think somebody started presenting in between, somebody else.
Got crashed. So in between that YouTube, there was a little hindrances. So I could I had to stop in between and I went again for the live session. So in between there is an interruption. So I think I will give the Google recordings to those who are interested. Apart from the
Okay.
YouTube recording, I'll send the Google recording also, Meet recording, to those who are interested. If you want to have the whole session, to know the whole session, I'll send that recordings to
Ma'am,
them.
I think we can maybe if we see that the YouTube has problems, this same thing can be uploaded again, right? The same Google recording
Yeah,
can be
Google
uploaded into
recording.
the.
Yeah, I'll record the Google thing and
And Sarin, I think you should mute all the people when the speaker is speaking.
yeah, muting. I had muted in between some those who were in the what you call the video mode and all. Uh, I muted them and I removed them also. So today actually there were 101 participants. So in between, if somebody got exited, they were not able to get into it, and some technical issues are there. But those were manageable.
Okay.
Okay.
Yeah.
Okay. So I think Google
YouTube
has limited to
also there would have been many people. YouTube, many people would have been there, no? Right?
yes. I think more than 20.
Ma'am, we'll have a clear picture by tomorrow. But I think our participation was maximum here in Google
Yes.
Meet.
Yes.
And once again, thank you, Sudhir sir. I really liked your presentation. How you managed to get in so many things
Yes.
at the
Yes.
same time. Definitely it is like, especially because I teach biochemistry, and these are some of the difficult topics to handle,
Yes. Yes.
especially plant biochemistry. So I found it a very convincing way.
Yes.
You started from the right from the beginning, the very simple way,
And the
and
pathways and
I,
all. Yeah.
yes, you led to the pathways, and exceptionally, I just like the way how you presented, sir. Thank you, sir.
Thank you.
Okay.
Okay, ma'am.
Okay.
Yeah.
Okay, bye.
Bye, ma'am. Bye, Professor Sareen. And
23rd, remember to attend, okay? Sudhir,
sure,
6:30.
ma'am. Sure,
Yes.
I
ma'am.
think 6:30, 23rd. It will be very interesting.
Yes.
CRISPR for genome editing
Genome
of
editing
plants.
plants. Yeah.
Sure, ma'am. I will.
I
Yeah.
will send you the link and all. Once it has been completed, I'll send you the
Please.
link, sir. In between ആരോ സംസാരിച്ചു എന്ന് പറയുന്നത് എന്താണ് ഫോണായിരുന്നു